This recording is part 1 of 4 for the cardiovascular system, blood vessels and circulation. Vascular transport is important in supporting life because it's how we distribute and facilitate exchanging gases, nutrients, and carbon dioxide and waste, as well as other substances with body cells. And anytime that vascular tissue is impaired, that can result in tissue damage in other areas of the body and also can impair metabolism. Remember, metabolism is the sum of all chemical reactions, as well as lead to system failure. So let's take a closer look at the cardiovascular system, blood vessels. In looking more closely at blood vessels, we notice that there are vessels known as arteries, which take blood away from the heart, arteries away, and there are blood vessels known as veins, which bring blood back toward the heart. There are also different circuits involved in the cardiovascular system. We have a systemic circuit, which carries oxygen-rich blood to body tissues. So it actually goes from the left side of the heart, the left ventricle, throughout body tissues, and then as our tissues use up oxygen, the oxygen-poor blood goes back toward the heart through veins to get to the right side of the heart, and specifically to the right atrium. And then we also have a pulmonary circuit where blood from the right ventricle is going to go towards the lungs so that carbon dioxide can be exchanged for more oxygen, and then that fresh oxygenated blood from the lungs comes back to the heart by way of pulmonary veins to the left atrium. In looking more closely at the structure of blood vessels, we have to get familiar with some terms. So the lumen within a blood vessel is that space internally, and the blood vessel walls consist of living cells and connective tissues. There are 3 distinct layers that are known as tunics. When we look at the tunics of the artery, we see that there are 3 layers: the tunica intima, the tunica media, and the tunica externa. And the same is true as with veins. However, in arteries, in the tunica intima, there's an inner endothelium, which is also true in a vein. There's a subendothelial layer, which is made up of connective tissue. And then in arteries, we have an internal elastic lamina, which is sometimes referred to as the internal elastic membrane. And that allows for stretch and recoil with pressure. With the tunica media in the artery, there is also an external elastic lamina or external elastic membrane. Again, veins do not have these layers. And then finally, both have the outermost layer, the tunica externa. So remember that veins just don't have that elastic layer, actually 2 elastic layers that are found in arteries. Here we have a picture of a model demonstrating the different layers of both arteries and veins. And you can also see in this particular picture, there is a valve in the vein on the far left side. That valve helps prevent blood from flowing backwards in veins. Here's a closer look at the tunica intima. Again, it's made up of simple squamous endothelium, and that endothelium is continuous throughout the vascular system. In fact, that's what ends up lining the inside of the heart. It's simple squamous epithelium that we call endothelium because endo means inside. And the endothelium, those cells, produce endothelins, which are peptides that can help constrict vessels. The basal lamina lies underneath the endothelium, and that helps bind it to the underlying connective tissue. And we see the internal elastic membrane, which is really seen in large arteries only. And then you can see the boundary between the tunica intima and the tunica media is basically formed by that internal elastic membrane. The tunica media is the middle layer of blood vessels, and it's made up of thick, smooth muscle cells and elastic connective tissue. It's important because it's responsible for maintaining blood pressure as well as blood flow, and it consists of mostly circular, smooth muscle cells and a little bit of longitudinal smooth muscle. This smooth muscle is responsible for vasoconstriction and vasodilation, and there are blood vessels and nerves that go to the tunica media and tunica adventitia, known as the vasa vasorum, which literally means vessel of vessels, and nervi vasorum. And that way there can be sympathetic stimulation for blood vessels to cause vasoconstriction, as well as a way to get oxygen and nutrients to these tissues and to get rid of carbon dioxide and wastes. We don't need this for the tunica intima, the internal layer, because it's got direct connection to blood as blood passes through the lumen. There's also an external elastic membrane or external elastic lamina, and that helps to separate the tunica media from the tunica externa, which again is also known as the tunica adventitia. The tunica externa or tunica adventitia is the outermost connective tissue layer surrounding blood vessels, and it's made up of mostly collagen with some elastic fibers. It is the thickest layer in veins and in some arteries. The outer layer is contiguous with other connective tissues within the body to help, where it courses through, to help stabilize those blood vessels. And you can see on the picture on the right how there are vasa vasorum and nervi vasorum as well in the tunica externa. That's how they get to the tunica media. They have to go through the tunica externa to get there. And I also wanted to point out in this picture, you'll notice the difference in the thickness of the smooth muscle tunica media between an artery and a vein. Veins have a much thinner wall of smooth muscle, and you can see that when you look at the picture on the right, or even the picture on the left where we've got the picture of the microscope slide. If you look at the artery below where it's got rings around the tunica media and rings around the adventitia, you can see the difference between that artery and then the vein to the right. It's got a much thinner wall because the tunica media is thinner. Large arteries, especially those coming right off of the heart, are known as elastic arteries or conducting arteries. If you think about the fact that the aorta is the first major vessel coming away from the heart, it's got to deal with high pressure as the ventricle, the left ventricle, has to generate a lot of high pressure to open up that aortic semilunar valve. And so they're known as elastic arteries because they have to stretch to deal with that high pressure. They're also sometimes known as conducting arteries because they're conducting blood away from the heart. They're the initial ones that do that, and they have a lot of elastic fibers and strong recoil, and they demonstrate compliance, which is that ability to stretch and recoil. So the largest arteries are elastic, and the ones near the heart are often sometimes called conducting arteries. As we go further away from the heart, the medium-sized arteries tend to be referred to as muscular arteries or distributing arteries, because they're going to be delivering blood to specific body organs. And they can also vasoconstrict, and that vasoconstriction helps to increase blood pressure if that's required. There is not as much of an ability for them to expand, so there's decreased elasticity or decreased compliance as we go further away from the heart. And there's no line of demarcation between conducting arteries and muscular arteries, so that's not something we're going to look for in the lab under the microscope. Anytime you see a word that ends in OLE or ULE or ELLE, that usually indicates a smaller version of that structure, and arterioles are smaller versions of arteries. Remember, arteries take blood away from the heart, and as they get further and further away, they do become smaller and smaller until they become arterioles, and arterioles will also start to become smaller and smaller as they go towards capillary beds. So arterioles are found between arteries and capillaries, and arterioles have very thin walls. However, they do have that circular smooth muscle in them, and then when they get down to that smallest version of themselves, they have smooth muscle cells wrapped around them. So the smooth muscle in and around these arterial walls is really important, because that regulates the diameter of the arteriole, which is going to determine how much resistance occurs within that vessel, and it provides vascular tone. So arterioles are actually the primary site of pressure regulation. This takes us to capillaries, and there are three flavors of capillaries. We have continuous capillaries, fenestrated capillaries, and sinusoid capillaries. We'll discuss all three. However, this slide is going to deal more with continuous capillaries. And capillaries are important because they allow for perfusion, which is the flow of blood to tissues, to occur. And the flow of blood through a capillary is known as microcirculation. Capillaries are the most vascular tissue in the human body, and they consist completely of endothelium that, of course, is going to be supported by the basement membrane. And continuous capillaries have tight junctions. Remember, tight junctions help keep things from going between cells. And in this continuous capillary, we notice that as those simple squamous cells overlap one another, that's where we see the tight junctions occurring. I'm going to circle that on this picture, and you can see that tight junction. And there are also transport vesicles for pinocytosis. The only place that does not occur is in the brain, and that's because continuous capillaries are the capillaries that we see that are found in the brain that help make up the blood-brain barrier. Tight junctions help keep things from leaking between cells, and blood is toxic to neurons, so we don't want blood to leak through, and that's why we have those tight junctions in the brain in continuous capillaries. And these are the least permeable type of capillaries, however, they are the most common. And again, this is what we see in the blood-brain barrier. When we look at fenestrated capillaries, fenestrations are pores. And so these pores are seen in the simple squamous cells themselves, and we still have those tight junctions that hold the overlaying or overlapping cells. So we've got tight junctions, but now we've got fenestrations. We also have those pinocytotic vesicles. Remember, pinocytosis is where the cell, they call it cell drinking, so that the cell can take in fluid, and the fenestrations are going to be permeable to larger molecules. So we see fenestrated capillaries in places like the small intestines, the kidneys, and the choroid plexus. So in the small intestines, things can come in for breaking molecules down for digestion. In the kidneys, we can move things in or out of the kidneys, and the choroid plexus, as you remember, in the brain is where we're going to produce cerebrospinal fluid. So the fenestrations in fenestrated capillaries help molecules move in and out depending on where they are. This takes us to sinusoidal or sinusoid capillaries, and these are the most permeable. We find them in places like the liver, bone marrow, in the spleen, as well as the adrenal medulla, and they've got larger intercellular clefts. So those we see that there are gaps between the simple squamous endothelium, and we also notice that there are larger fenestrations and a few tight junctions. The few tight junctions that do exist help hold the simple squamous endothelial cells together. And the sinusoidal capillaries tend to be more irregularly shaped, so their path is more tortuous as far as how the blood flows through, and they have larger lumens. Blood tends to flow slowly through them, and there are also macrophages that can actually reach through the clefts to catch their prey on or in the liver, where they form part of that wall of the sinusoid. So they can actually kind of reach through and grab stuff. In certain places in the body, arterioles may form structures known as medarterioles, which are similar to both arterioles and capillaries. Medarterioles consist of a tunica media, which is discontinuous, but also has rings of smooth muscle cells around it, so that those can act kind of like sphincter muscles. The sphincter muscle is a circular muscle that can close a structure down. And medarterioles lead to capillary beds. Capillary beds can consist of, oh golly, anywhere between 10 and 100 capillaries. So what we see is the precapillary sphincters, depending on circumstances, may be stimulated to contract, in which they'll constrict and shut off blood flow to the capillary bed in order to direct blood flow elsewhere. A really good example of this would be in a sympathetic nervous system event. You would not necessarily want perfusion through the digestive system when skeletal muscle tissue needs more blood flow so that a person can run away from a bad situation. And that's, again, going to be an area where capillary beds may not receive as much blood flow, and instead the blood would be directed through an arteriovenous anastomosis, where blood, instead of going through that capillary bed, will go through the arteriovenous anastomosis, which is always going to be open, but if, again, in a sympathetic nervous system event, the capillary bed is going to experience a lot less perfusion and more blood goes through that anastomosis. And you remember an anastomosis is just a joining of vessels. When blood flows through the capillary bed, they refer to that motion of blood flow through a capillary bed as vasomotion, and it's not very smooth. It's actually very kind of slow and pulsating. The blood, as it flows through capillary beds, has the ability to allow for diapedesis, which is where white blood cells can actually migrate through the walls of the capillaries into surrounding tissues. And this would be important if there was an infection. Again, perfusion is really more likely to occur wherever there are capillaries and large capillary beds, as long as those sphincter muscles are not constricting and shunting the blood or directing the blood elsewhere. From capillaries and capillary beds, blood will next flow into venules, which are smaller versions of veins. As blood flows from arterioles or meta-arterioles through to capillaries or capillary beds, or sometimes through an arteriovenous shunt, the blood next goes into venules, which are small versions of veins. So these are post-capillary, after the capillary structures, for the most part, unless it's from an arteriovenous anastomosis. And multiple venules will join to form a vein, and more veins will join together to form larger veins. And veins are going back toward the heart. When we look at a venule, these are very small structures. They consist of endothelium and a thin layer of smooth muscle, as well as a layer of connective tissue. And diapedesis, which is that motion of white blood cells out of a vessel to surrounding tissues, can also occur in venules. When we look at veins, veins are, again, still going towards the heart, and they consist of thin walls. They have larger, irregularly shaped lumens in comparison to venules. The pressure inside a vein is going to be lower, and so veins have valves that allow for blood to flow in one direction toward the heart. Remember that blood from the lower portion of the body has to flow against gravity, so we don't want it falling backward. We want to keep moving it up towards the heart. And so skeletal muscles that surround blood vessels can help that return of venous blood flow to the heart when they contract. When the skeletal muscles surrounding blood vessels contract, they actually help move blood back toward the heart. And if we look at this picture on the lower left-hand side, you can see the thicker walls of the artery, and then the veins have been outlined in blue, and they're thinner walled. Because the wall is thin, it doesn't have that nice round structure to it. The muscle just doesn't give it the support that the arteries get. with a large vein. You'll notice on the right-hand side of this PowerPoint that larger veins, what you can't really tell is their tunica media is, it's not that it's smaller than a medium-sized vein, it's just that that whole vessel is larger, so the tunica media appears smaller. But in medium-sized and large veins, we have, again, the tunica intima, the tunica media, the tunica externa. What we don't have are the sheets of elastic connective tissue that we see in the arteries. And we also see the vasa vasorum and the nervi vasorum in the connective tissue surrounding the veins, so that those structures can get to the tunica media. Because it is smooth muscle, it does need a blood supply to perform its actions and nerve innervation as well. There's a concept with which you may not yet be familiar, and that is the concept of interstitial fluid. That is the fluid that surrounds the cells throughout the body, excluding what's in the blood vessels. And that's the interstitial space, is the area around body cells. And again, it's a fluid-filled space, so we refer to that fluid surrounding our body cells, which is also referred to as extracellular fluid. That's also interstitial fluid. And so if blood is pooling for any particular reason, maybe there's excessive blood due to hypertension, heart failure, or renal failure, where blood is collecting in an area, or there's a lot of pressure forcing blood through smaller vessels, that can lead to some of the fluid portion, the plasma, to leak through those vessels, causing an increase in interstitial fluid. And if that increase in interstitial fluid is great enough, it can cause what we call pitting edema. And you can see how pitting edema is rated on this slide on the left-hand side. And you can see a picture of it above me. And again, pitting edema is just a symptom, but it could be due to either excessive force in the blood vessels from hypertension. It could be due to heart failure, where blood backs up and starts to pool, or kidney failure. We talked about the fact that veins have special structures known as valves. And if for some reason those valves should become defective, then that can produce what we call varicose veins, which when the valves fail, the blood tends to accumulate on a valve distal to it, and so we see swelling and we see tortuous veins starting to occur, because the blood is just not being moved up towards the heart. So in a varicose vein, if the valves fail, they're just not closing properly. And again, that can allow blood to pool and cause the vein to distend or enlarge. If it's really bad, it can lead to ulceration of the skin because of the pressure of that blood vessel on the skin. It can actually cause pressure necrosis or tissue death. So these can be very painful. They can be addressed surgically. And if they're really bad, they need to be addressed surgically. Our veins are actually responsible for acting as blood reservoirs. And if we look at systemic circulation, not pulmonary circulation and not what's in the heart, systemic circulation is where we find about 84% of our blood at any given time. And within that 84%, our systemic veins have 64% of that volume. And so you can see those percentages and how they break down. Our systemic arteries only have about 13% of the systemic circulation. So here in this slide, you can also see the difference between an artery, which is in the red circle to the right, and also in the red circle to the left, and a vein, which we are seeing lengthwise. Actually, for both the artery and the vein, it's just the way that things were cut. And so you see the vein in blue, and they're just showing you the endothelial cells in this particular microscope slide.